Morning.
Morning.
The enthusiasm is just overwhelming. Good morning. Come on, I was trying to make an espresso, but you guys logged in too early, so I'll have to wait till we're done. How's everybody?
Good.
Good. Okay. All right. So we actually had some really neat questions last week and some back and forth on a number of different topics. And so I emailed Mr. Somerville, not last email, some suggestions, but he followed up and said that somewhere that you had some discussion following the session. So do we have some questions that grew out of that we should just follow on with?
Juliana.
Yes. So who wants to go first?
I'll go. So I was just wondering, if you sever a nerve in your leg, how come the nerves will not grow back, allowing your leg to work again?
The evil twists of the body. So that's a really interesting question. And it sort of comes out of the regeneration question that we had last week. And so, okay, just keep in mind, I'm a fly geneticist. So as soon as I start talking about developmental neurobiology, there's a chance I'm making this all up. So you should take this with a grain of salt. But it is a really interesting question. In humans, we don't have a lot of neuroregeneration. And in other species, we do. And I don't know that we have a great handle on evolutionarily why neuroregeneration works better. So, when a salamander regrows its arm, it's a fully innervated arm. When I was an undergraduate, so in the 1980s, there was a lot of discussion on whether there was any regeneration in the nervous system. And the conventional wisdom was there was not. And I think this came out of a lot of sort of brain injury research. But there was really interesting work that was done with songbirds. Can anybody guess, from what you know about songbirds, and this is just what every school kid knows about songbirds, why would songbirds be an interesting model for neuroregeneration? What do songbirds do on a seasonal basis? Anybody? Bueller?
Migrate.
They do migrate. Actually, that's a better answer than the one that I was going to come up with, but it's not migration that we were interested in. So people were looking at songbirds because male songbirds call in a very specific way to females for a very brief period of time. And so during the mating season, many species or many males of species of songbirds will call to the female in a mating song. And what we realized in the 80s, 1980s, maybe 1990s, was that the region of the brain that controlled mating song got bigger in males during the mating season, and then it got smaller. And we realized this was a really interesting case of growth and death in the nervous system, and that's been developed as a model system for how neurological systems can change and can regrow. So this comes out of the observation that after injury, we don't get a lot of recovery of the nervous system in the same way that we get recovery of the muscular system. And we're still struggling to figure out why. The simple answer is probably that the nervous system is really complex. And because it's complex, it has basically evolved in such a way that it develops once, and it's very difficult to redevelop. But that can't be the whole answer, because in some cases, like salamanders regenerating arms or songbird regions of the brain expanding and contracting, we do get regeneration. So the shortest answer is we don't know, but there's a lot of really interesting work going on looking at that. From my perspective as a geneticist, genetics plays a big role here, and there's something that doesn't allow those genes that are involved in neurodevelopment to be turned on again to regenerate those nerves. So does that start to get at your question?
Yeah.
So if it's something you're interested in, take a look at the songbird stuff. It's something that I found really fascinating when I first went to university. And it's a really neat way that people have realized that we can study something very effectively. And there are other things out there where we use models in mammals where you can go in, and in rats, for example, there's a nerve in the leg that they cut very often in research and look at that. But the songbird brain stuff has been really fascinating about what it tells us in neuroregeneration. And there's a lot of interest in that with brain injuries and with aging, so deterioration due to dementia or Alzheimer's. And we've been looking for alternative models to study that. All right, that's a great question. Do we have other great questions that grew out of last week's discussion?
Someone else want to go? Okay, I'll ask a question. Are some genetic traits more hereditary than others?
Yes. So, pretty much any trait, and there's actually a whole school of research that defines just how heritable a trait is, and we define it as heritability, and even within what we call heritability, there's broad sense heritability and narrow sense heritability, and there are different ways that we think about how traits are inherited. So any trait basically is a function of three components. There's genetics, the environment, and random chance. And some traits, that genetic component is the majority of what determines what the trait is, and in other traits, it's a very small minority. In traits that we call simple traits, so simple traits are Mendelian traits, so things like pea color and pea shape, Mendel's round peas versus wrinkled peas, yellow peas versus green peas. There, the heritability is really strong, but it's still not 100%. There's still a large genetic component. There's still a component. While there is a large genetic component, there is still a large component of random chance and the environment. Most traits are what we call complex traits, and most traits that we're interested in, things that we talked about last week, like intelligence, those are complex traits. Even within relatively simple things like cancers, different cancers have different degrees of heritability. Can anybody speculate or guess why different cancers would be more or less genetically controlled, would have greater or lesser heritability? What have we talked about in driving cancer? There's the one central thing that drives cancer. We're going to remember what that one piece was. All right, good. So everybody else needs a coffee. So I'm going to take a break, and I'm going to go get my espresso, and everybody else can caffeinate, and we'll sort of liven the discussion up. Okay, the one thing that is common in all of these cancers was gene expression. Right? So it's the wrong gene being turned on at the wrong time. Different cancers are going to have different genes that are involved with them. And one of the reasons that we have different heritabilities is the way that these genes are regulated. So some genes, it's a really simple genetic switch, and other genes are really susceptible to changes in their expression from changes in the environment. And so that gets us to this idea of why there are. So the answer is, are there different heritabilities, different amounts of heritability? Yes. And then scientifically, the really interesting question is, what's driving those differences? And what seems to be driving those differences is the kind of genes that are involved in the traits that we're looking at. All right, does that get us started on your answer?
Yes, it does. Thank you.
Okay, you're welcome. All right, everybody staring at me. Somebody stare at their paper and come up with a question.
Come on, folks.
Okay, what is the chance of someone getting cancer if they keep their phone on in their pocket or if they put a computer on their lap?
I don't even have a snarky answer to that one. I'm not sure where we're coming from on that. So is there a reason that you are putting a connection between a phone in a pocket and cancer?
Well, I'm just curious because I heard that it can cause cancer because of the radiation.
Okay. I haven't heard that. That's interesting. I haven't heard that one in particular. I don't know the amount of radiation that comes out of something like an iPhone or a smartphone. I know that there's been a lot of speculation about things like brain cancer from the amount of time that we spend with the phone stuck up to your ear. As far as I know, there's nothing that actually shows that connection. But I don't know that we know the answer to that. The issue is coming from basically this idea of radiation, and we know that radiation is a carcinogen. So then the question is how much radiation is coming out of these different devices that we're working on. There was another big thing that was in the news. Actually, it's been a little while ago now, but it was a big deal for a while. Has anybody heard about the issues around power lines? So there was a similar speculation that these high power lines that run through a lot of southern Ontario, we know that they throw off large electric fields. And there was a concern about what these electric fields were doing to the environment. And there was a proposal that they were causing cancers in people that were living underneath power lines. I don't know what was ever resolved of that. One of the issues that comes up with a lot of these broad studies of cancer is they end up being sort of anecdotal. And somebody knows somebody whose friend heard of somebody that lived under a power line and they got a brain tumor. And suddenly that becomes evidence that power lines cause brain tumors. And this one actually made the media circuit for a while, and I know there were actually some fairly high-profile scientists involved, maybe on both sides, trying to figure that out. So if there is a danger to cell phone use or just carrying around a cell phone and or a notebook, it's going to be related probably to the electric fields and radiation that are emitted by those devices. And the question is, how do we test that? And that should be relatively straightforward. I think it'll be interesting to find out in the long run whether that pans out. For quite some time, there was a lot of hesitation to accept that smoking caused cancer. And in the 1950s, you've probably seen these ads. There were a lot of ads talking about the health benefits of smoking. Ronald Reagan, the former American president, famously did some print commercials in the 50s where they were touting the health benefits of regular smoking. And now I don't think anybody doubts that smoking causes cancer. So I would be surprised if cell phones caused cancer, but I wouldn't be surprised if something that we're doing on a regular basis is having the same kind of adverse health effects that the cancer has, or that smoking has. So that's an interesting question. I don't think we have a great answer to it at the moment.
Okay.
The group step up.
I have a question that's not really related to any of the previous questions already asked, but at the moment we just finished our optics unit and we're writing a lab report about our final project. So I was wondering what kinds of lab reports you've written and what you should focus on in the conclusion portion of the lab report.
That's an awesome question. And this is an area that we have a lot of work to do in education. How do we train people in technical writing? And it's one of the hardest. So communication is one of the hardest things to teach. And Mr. Somerville probably knows, or almost certainly knows, a lot more about this than I do. Communication skills are one of the most important things you can get out of any class that you're taking. When you're taking a lab class, you're often writing these lab reports in what is essentially a very artificial format. And one of the things that we've tried to move towards, at least in the upper level courses at university, is getting the students to write in a format that makes sense professionally. So what I'm going to do when I finish today, I'm going to have lunch, to be honest. And then I'm going to go for a paddle on my paddleboard because it's the summer and I deserve to go for a paddle on my paddleboard. After I've had lunch and gone for a paddle on my paddleboard, I'm going to work on a paper that I just accepted in a journal day before yesterday. And it was accepted with minor revisions, so I have to go back and edit those revisions. So that's a lot of what I do as a scientist is write up my research. I get a lot of federal money to do research, and I promise you I will get to your question. It's a roundabout, but we'll get there. I get a lot of money to do research as a scientist. My obligation to you, or at least your parents at the moment, since they're the one actually paying the taxes, is to communicate those results. And so one of the things I do is chat with groups like this about what it is to be a scientist and what our results are. But a big part of what I do is I write scientific reports. And a big part of what I do as a professor is teach my students to write scientific reports. When we're writing those reports, the first thing that we write is the methods. And in theory, you know what the methods are and you know what you did, and you are working now on just how to communicate that. So it's not a question of what methods to include, although surprisingly that does become a question fairly often, but it's how do you clearly communicate to the reader what those methods are. So if I'm doing that, what do I need to get across to you as a reader in a methods section? What do you need to know? Or what do you need to get out of that methods section? Anybody? Come on, this is an easy one. They only get harder from here. If you're reading the methods section of a paper, what do you need to get out of reading that section?
You need to understand how to do the experiment?
Yes. And actually, that's maybe the second thing. The first thing is you need to understand what I did. And you need to be comfortable with what it is I did in that experiment, because that's going to be your basis on whether you believe anything else that I've done. So in that methods section, I have to explain to you what I did so that you can decide for yourself whether the rest of my experiment, whether the rest of my report has any credibility. Okay, the second thing was your answer. Oftentimes, I read a paper because I want to either duplicate those results or I want to use that technique in another question that I'm asking. So as a reader, I have to be able to get enough information from that methods to really understand what was done. So when I'm teaching somebody to write, the first thing we do is write the methods. And you can often write the methods while you're still doing the results and you're doing the experiments. Okay, the next thing that we do is we write the results section. And really the first part of writing results is what? Can anybody guess? This is where I'm going to put the information in that report. What do I start with when I write the results?
Your hypothesis?
Nothing. No. So we're going to come back to hypothesis, and I got to tell you, I'm really terrible about doing hypothesis-driven science, but that would be one of the things. In the results, the first thing that I have my students do, and I'm looking for an example, and I'm not finding one, the first thing I do when I write the results is I have my students make the graphs. So we draw the pictures. And we as people are very visual. When we're writing a results section, the first thing we do is we plot the data out. We make graphs. And we make more graphs than we could possibly put in that paper because I want to be looking at the data and trying to process that data in my head. So we write the methods, and then we draw all the results, figures, the graphs, the tables, everything in some sort of visual format so that I can start to process it. Once we have those figures, those tables, those graphs, then we go back and we write the results section. What does the results section have to do? What does the reader need to get out of that results section?
The conclusion. What you concluded from doing this experiment.
We're going to get to the conclusion, but that's actually the next piece. What do you need to have to get to those conclusions?
Data?
Sorry?
Data?
Yeah, so data. What I have to get across in those graphs is what the results really were. What kind of data did we collect? What are the numbers? How many flies did we assay? How did we assay them? What are the trends in the data? And that results section, then, so as a reader, you have to understand what did we find. And from that, we're going to develop the conclusions. So you asked, what do we put in the conclusion section of the paper, I think. That's the third step. So We have to be clear about what we did in the experiment. Then we have to be clear on what the results are and the trends that we think we're seeing in the results. And that's the point of the results section, is arguing to the reader, this is what we think we see in the data, and this is why we think you should believe us. So in that methods and the results section, you're trying to establish with the reader your credibility so that when you state your conclusions, they know whether they can agree with them. Does that make sense?
Yeah.
And one of the tricky things in the conclusion section is balancing what you would like to say with what the data actually says. So we often go into a project trying to answer some large question. And with the experiments that we do and the results that we get and the data that we actually collect, we're able to answer a smaller question. And from that smaller question, we can extrapolate out to larger conclusions. But you have to make the justification in your writing for that extrapolation. And if you simply say, we weighed this rock and the rock weighed 4 kilos, that may literally be true, but it's not an interesting conclusion. If you can say that we weighed this rock and we weighed this rock after treatment with some sort of erosion solution, and what you're trying to do is quantify just how much that solution eroded away the rock, you can say the rock weighed 2 kilos less. Fine. That's literally true, but it doesn't tell you anything. If you can say, because of the process of erosion, we lost 50% of the mass of the rock, and this suggests that in a natural situation, we're going to have this effect of erosion. Now you're getting into interesting conclusions and broad statements. That's where it becomes really informative. So in the genetics work that we do, we'll ask really small questions that we can get discrete, finite answers to, because we want to answer larger questions about how gene regulation works overall. So in the paper that I'm going to be working on the edits for this afternoon, there are a couple of points where the editor said, look, I know what you want to say is this, but literally your data really only supports this, and you need to either justify this extra step or explain that it's a leap of faith and why you think that's justified. So that's a lot of what I'll do this afternoon, is working on how do I go from what I really want to say to what the editor agrees with me that I can literally say from the data that we have at hand. And that's a big part of learning how to communicate the results that you have. All right, does that get to your question about conclusions in the paper?
It does, thank you.
Is it really? Because I might have just sort of talked around it. Because
it made sense.
We good? We're good? Okay.
Yeah,
Great. Anybody else?
Yeah. Here,
Okay.
question. Okay. So after you finish the conclusion part, now we can get back to the hypothesis. How do you write like a good hypothesis, or like how should a professional hypothesis sound like?
Oh, yeah, you need to talk to my wife. My wife is really awesome about hypothesis-driven science. Unfortunately, I'm more of a, I don't know, let's try it scientist. And so I end up doing these post hoc hypotheses. It's obviously a really good question if I'm this stumped by it. But the way that we try to do science in my lab group is we come up with broad picture questions that we're interested in. And then we put together smaller testable questions. And these are, I guess, technically the hypotheses. And so we'll generate a question that we can address experimentally. And the trick is to make sure that the experiment is designed well enough that no matter what the results are that you get from that experiment, it tells you something about the question. So a common pitfall in the way that we ask questions in science is you design an experiment, and as long as you get result A, you can interpret it. But if you get result B, that could be because of explanation 1, 2, 3, 4, and you can't distinguish those. So you do the experiment, and if you get result A, then fine, you can draw this conclusion. But if you get result B, you haven't gone anywhere. So one of the things that I try to teach my students is how to design experiments such that no matter what the results are, they give you some part of the answer to the question that you're trying to address. So I know that's not exactly the question that you're getting at, but as you start to ask experimental questions, you want to keep in mind the big picture questions, but you can almost never answer an entire big picture question with a single experiment. So you then design experiments that will allow you to answer the smaller questions that you can put together to start to answer the bigger questions. That might help. Does that get anywhere towards answering your question?
Yes.
I think the answer then is no. Okay. Do you want to rephrase your question and see if I can pound at it from a different direction?
Like the format of a hypothesis?
Oh, yeah. I'm the wrong guy to ask. I'll be perfectly honest with you. I still struggle with this, and I'm a tenured full professor, and I've been doing science for 20 years. There are definitions on what counts as a hypothesis versus one thing or another, and I'm pretty terrible at that. But I can tell you that you can actually be a reasonably good scientist and still be pretty terrible at defining what a hypothesis is. So while I can't answer your question, I can tell you that even if you don't get an answer, it doesn't mean you don't have a future in science.
One more.
All right, I think this might be our last session. So does anybody have any questions just about university, about being a professor, about being a scientist, sort of broader questions beyond? I mean, and if you've got any more biological questions, as long as it's not defining a hypothesis, I'll at least give it a shot.
Define a theory then.
Yeah, semantics is not my strong point. I'd have to make something up. You'd be much better off. Sorry?
Oh, I was totally joking. No, don't give it up.
Okay, thank you.
What kind of fish is that behind you?
It's a South American cichlid. The common name is a Midas. And when I bought it in 2006, it was this big. And it was mislabeled as another cichlid, and so I brought it home with its pair and a couple other fish, and it is about 10 times bigger than it was, and it killed everything else in the tank. They're very big and very interactive, and they're horrible aquarium fish because they don't get along with anything. All right, so are you wrapping up? Does your session end tomorrow?
Yes, it does.
Yeah. Okay, that's a quick summary.
I think Emma has a question.
Yeah. I just have one more quick question, I guess. So I think it was mentioned earlier that we're writing a lab report right now, and this is kind of the first time that our teacher isn't providing us with any analysis questions. I was wondering if you have any advice on what sort of questions we should be answering in our analysis.
All right, so you've already done the experiment?
Yes, we have.
Okay. There's the short answer is you should always know the questions that you're asking before you do the experiment. And then the real answer is nobody always knows the questions that you can ask until you do the experiment. But the point that I made earlier about graphing out the data is entirely correct. And I found, or I realized the value of graphing data when I was a postdoc. So through my entire PhD, the people I interacted with kept saying, graph the data, graph the data, graph the data. So I would draw all these pictures, and I literally would tape them, I'd print them out, or scribble them on notepaper, and then I would tape them on the wall above my desk, and I would just stare at these things. And it ends up, that's actually a really effective way to get at what your experiments can tell you. And when I was a postdoc, I'd done some experiments, and I just put the results in a table, and we ended up publishing the paper. But I went back a year later to make a presentation to give as part of a lecture, and instead of making a table, I graphed the data. And when we had been doing this experiment, it was some very basic biochemistry. We'd taken 2 alleles of an enzyme that I work on in fruit flies, and we had looked at the Michaelis-Menten constant, which is essentially, it's an indicator of how tightly an enzyme binds its substrate. And based on an amino acid change between these 2 alleles, we had hypothesized that one of the alleles would have different binding kinetics than the other, and we actually had a prediction as to which one would bind most strongly. And statistically, there was no difference in the alleles, and so we left it at that and said, while we had proposed that there was a difference based on this amino acid change, there was no statistically significant difference, and so we were probably wrong. When I graphed the data, I realized that the trend was exactly the trend that we had expected, and the error just slightly overlapped. And if I had graphed this data when I did the experiment, I would have realized that basically I had too small a sample size. And if I had done the experiment again with more flies, I would likely have gotten the answer that we predicted based on this amino acid change. And I missed it because I never graphed the data. I just looked at the numbers, and my brain in particular, but the human brain really in general, is not great at processing a page full of numbers or a page full of letters, but we do a really good job with figures. So when you're trying to figure out the kind of questions that you can ask, take a look at the experiment that you did, graph out the results that you have, draw cartoons of the graphs that you have, and try to get a feel for really what can you say about the data that you've generated. You sometimes will be surprised how much more your brain will start to process. Now, the other side of that is your brain will make stuff up. And this is the reason that we have statistics, is your brain will start to see patterns because your brain is genetically programmed, essentially, to find patterns. So you look at the data, you try to find the patterns, and then you go back and statistically test that and say, okay, do the numbers really support that? But oftentimes the interesting conclusions are, we see this pattern in the data. It's not statistically significant, but the pattern suggests that if we did the experiment again with a larger sample size, this would be an interesting result or interesting conclusion. And that's often the way that we do science in my lab. We do the experiment, we look at the results, we realize what's interesting, what's exciting, and what we screwed up, and then we do it again with more replication or better controls. I think we're probably running out of time, but let me give you one last really cool example. The paper that I'm going to work on this afternoon. So I published a paper a number of years ago that was worked by a graduate student where he showed a particular pattern in these deletions that we had created in the fly genome. And I had a new student join the lab, and David went back and he replicated all of Thomas's work, the first grad student. And when he did that, he didn't get the same numbers. And his first assumption, or at least my first assumption, was that David had screwed something up. He was a new student in the lab, and he just couldn't get the experiment to work. But when we looked at the data, it suggested that there were interesting variables in the system that we hadn't anticipated. And what we did was go back and redo the experiment, but now control the environment in a way that we hadn't done in the first experiment. And what we found was an enormous environmental effect about this genetic trait. So the whole point of the paper today that I'm going to work on is that this particular trait that's an interaction between chromosomes that drives gene regulation, that interaction has an enormous genetic or environmental component. And we only look to study that environmental component because the student redid an experiment. We graphed the results of one experiment against the results of the other experiment, and they didn't match up. And that made us go back and do the experiment again with different controls. And that suggested very different questions than we'd ever guessed. Nobody was thinking about this particular kind of genetic interaction in an environmental context. And because we looked at this data, we started thinking, shit, I think environment's a really big deal. And now we've gone back and we've reanalyzed people's results from other papers. and shown that once you start thinking about environment, it allows you to explain other people's results more thoroughly in things that they'd had to sort of hand wave. Like, well, we're not really sure why there's a difference here. The difference is because there's a difference in environment. So look at the data that you've got, look at the experiments you've got, graph things out, try to find patterns, and that hopefully will direct you towards the kind of questions that you can ask.
Awesome.
Thank you.
All right, I think we've taken up enough of Dr. Merritt's time.
Yeah, I've got lunch and paddleboarding coming up. I gotta rush.
Do you have to break the ice on the lake before you go out? Or how does that work?
I'm not allowed to give you the finger, right? Is that one of the things that I'm not allowed? So I will say that we were almost 3 weeks late icing out this year. And I literally could not row because the lake was still frozen. So yes, while I know you were teasing me, that is unfortunately very true. Folks, this has been awesome. Thank you so much for chatting with me.
Thank you. Great.
Enjoy the rest of your summer. Take care.
Thank you. Bye-bye.
Goodbye.
Bye.